Okay, thank you. Good morning, guys. So my name is Rhonda, and I use she, her pronouns. I'm coming to BeeCon from Dr. Leonard Foster's lab and the University of British Columbia, which is located on the traditional, ancestral, and unceded territory of the Musqueam people. Today, my talk is on my master's project, looking at the major determinants affecting honeybee health in British Columbia's blueberry crops. And before I get started, I'd just like to preface that this project began last winter. So although we have collected all of our samples, I don't have any results to share. So I'm going to be mainly talking about background and methods with you all. So honeybees and their hives are, as we all know, routinely placed in flowering crops during Canadian summers to pollinate them. And this fertilization allows for growth of seed or fruit. And again, as this crowd knows, blueberries are actually a notoriously difficult forage source for honeybees. The opening of the bell-shaped flower is usually very narrow, and that's too narrow for honeybees to efficiently access the anthers, which means that they collect much less pollen from the plants. Numerous native bees, including bumblebees and solitary bees, are indigenous pollinators of blueberry plants in North America. In addition, honeybees are used extensively by growers to augment these populations of native pollinators, kind of to make up for it. So maintaining this blueberry pollination and economy relationship is very important, as BC is responsible for growing 95% of Canadian blueberries, and this national industry itself is worth $250 million per year. Recently, though, I'm not sure where that didn't come up. Awesome. Okay. Recently, though, BC beekeepers have noticed a decrease in health and strength of their colonies following blueberry pollination, leading some to avoid the pollination contracts with farmers as it becomes financially difficult to sustain these hive losses. And even though this has been an outstanding issue for the past few years, the risk factors affecting bee health in highbush blueberry pollination are not yet well defined, nor have we really found a way for the stressors to be rapidly detected. One major reason why these stressors aren't very clear yet is because they may be acting alone or collectively to disrupt hives. And there are many different forms of stressors out there. Just like any other organism, honeybees are complex and they are also subject to multiple external risk factors that are going to make them more vulnerable to disease. So like I just mentioned, there are a lot of potential health disruptors out there that the hives could be facing during their interactions with blueberry flowers. And these stressors may be acting alone or collectively. So this is what we like to call the 3 P's of honeybee health. So we have pesticides, pathogens, and poor nutrition. And then subcategorized are the most common stressors that we look for. So this project is actually going to focus on pesticides and pathogens, at least initially, as most studies out there on blueberry pollination, specifically for honeybees, have not really emphasized that blueberry pollen or nectar is really an effector or a stressor. So pathogens and pesticides, on the other hand, are more variable external factors that could be present due to differences in crop management practices, as well as the interaction of other organisms with flowers. So currently, the honey industry relies on post-mortem analysis to test for the presence of a few known pathogens or toxins in dead colonies. Omic approaches, however, have become very convenient for observing changes between healthy and diseased systems because they allow for a fast diagnosis on a very small sample. So our lab at UBC focuses on proteomics, where we explore all the proteins produced by a single organism, and in this case, honeybees. So exploring the proteome is in a project like this is justified by the fact that the proteome is very central to health and its composition is likely to vary with health status. So the advantage of exploring the proteome is we get this complete picture of multiple stressors, which is a single analysis on a relatively small sample. For example, like in this corner figure here, 1 tube per hive is enough. So to investigate proteins, we use mass spectroscopy technology. There's a traditional type called data-dependent acquisition, which you'll see on the left side of your screen. It's DDA, and a newer type called data-independent acquisition, DIA, that we are actually going to be taking advantage of for this project. So with the older method, the second mass spec scan of the sample is completely reliant on what's found in the initial or first scan, as you can kind of see in that image. Whereas the new method accounts for everything in the sample initially. So what's really cool here is that by using DIA, we aren't biasing our samples anymore. It might be a little bit more computationally extensive, but we lose the headache that kind of comes with this inconsistent and sometimes maybe lost data. So the question that I am really asking here is which stressors are affecting honeybee health in blueberries? And I'm starting to explore this question using two methods. So first by sampling bees in the field and then doing proteomics on them, and then taking that field data into the lab to do these controlled cage experiments to find out cause and effect. So first we went into the fields where the goal was to quantify the natural variation or field realistic pesticides and pathogen stressors experienced by hives near typical blueberry agriculture in British Columbia's lower mainland. So these field experiments were done over 2 seasons in 2020 and 2021, and they're now finished. So what we did was we placed a bunch of hives in and away from blueberry fields. We then sampled all the hives before going into their sites at peak blueberry bloom and then following blueberry pollination. So this timeline was the same for the designated unexposed hives, which were also moved to very specific sites that were kept a minimum distance of 1.5 kilometers away from crops. The ideal distance for these unexposed hives would be greater than 5 kilometers away from many crops. So we had some variation, but always maintaining the fact that they were unexposed. And then we sampled nurse bees, which were run through proteomics. And that proteomic data from these field bees actually will determine the most common pesticides and pathogens present in these BC blueberry crops, as well as the doses that our cage trials will be exposed to. So going forward, talking about these cage trials, we're planning on doing cage experiments using these most common pesticides and pathogens discovered in our field trials. So these cage experiments are going to allow us to establish cause and effect, which we really need to validate our data. So here the bees are going to feed on a sugar syrup solution for 24 hours that contains pesticide and pathogen doses before I run them through proteomic analysis. And then statistical comparisons between control and exposed or treated bees will then be used to define the protein markers affecting bee health. So I want to take a second to emphasize that our sample set is nurse bees. So this means that in the field and in cage experiments, we are specifically looking to collect nurse bees who have interacted with the products collected from blueberry crops, so that our results are as consistent and as realistic as possible. So some of you might be a little bit curious about how we go about analyzing protein from a whole bee, and the answer is we don't. Instead, we dissect our bees for specific tissues or tissue. In my case, it's plural, that we anticipate to be affected. So we dissect for specific tissues so that we get accurate readings to explore our stressors. So I have initially kept my investigation very general by dissecting for the head, the intestine, and then the abdomen without intestine. And a really cool thing to note here is that the same protocol could be used to investigate other tissues of interest from the honeybee, such as hemolymph or fat body, which I might get into later on in my thesis. I'm kind of hoping to. But before we kind of load those tissues onto our mass spec, we need to clean up these samples to make sure that we only have protein, and that's the only thing in the resulting pattern. So taken all together, the vision of this project is to document the stressors honeybees face during blueberry pollination using proteomics. We'll use these biomarkers to understand how stressors impact colony health, and increasing honeybee health during and after blueberry pollination will hopefully ensure a sustainable and strong supply of colonies, which is essential for high blueberry crop productivity in BC. So briefly, just taking a look at where we're at now with this project. So we just came off a really enjoyable and successful season collecting nurse bees from our exposed and unexposed sites. All of our bees have been dissected by some really great undergrads and my high school student and myself into their 3 tissues. And they're all currently waiting patiently in our -70 freezer as we continue to optimize our proteomics protocol so that we can make sure that we do a really good job on that. So we are optimistic that sample processing will eventually begin this November. And yeah, I just want to say a huge thank you to BeeCon and the Center for Bee Ecology, Evolution, and Conservation for having me talk today. I'd also like to thank all these amazing people for their help on the project, and also to the BCSI team, which Sarah talked about, for sharing bits of this project with me.
Cool. I think there's time for one question, and I saw one in the chat. So basically, Chris asks, why are blueberry farmers relying on EHBs for pollination? Aren't native bees more effective?
Yes, Chris, you are definitely right. So native bees are much more effective. You should check out, there's a paper from NC State about this, where they compete, and SFU actually, where they compared 3 types or 3 species of bees for blueberry pollination, and you're 100% correct. The huge draw for blueberry farmers to kind of sign on to these colony contracts with honeybees is basically kind of just like a management opportunity, and to get full confidence that their crops are going to be pollinated. But you're 100% right. Honeybees are only, I think the statistic is 15% as efficient compared to bombus species.